There you go.
Actually, we have to change it again because it's going to be on the...
Let's change it back to
Yeah. Sorry.
to this.
Yeah.
We're good?
Yeah.
Sure? Okay. So look, Virchow did, he did an experiment. What they did is they put some food or something, which is non-living, left it in a test tube or a beaker for 2 days. And then the scientists said, whoa, lives come from non-living things. Food can give you rise to bacteria, to fungus, or to flies and stuff like that. It was actually flies. Even the music agrees with me. So, and then they start publishing papers. This was, I think, in England a few hundred years ago or something like that. Virchow was like, you know what, let me do one more thing. So what he did is he took a test tube, a flask actually, that was closed, and then what he did is he made a flask that has a long tube, as long as there's air coming in. And then he put in here like food. So food got oxidized, got, rotten and so forth. But there was no growth of living things. So he concluded that the growth that occurred on that food must have been introduction of bacteria or flies or something did that. So that after that, everybody started believing until today that non-living thing cannot give rise to living things. Cells cannot come from non-cellular things. And an example is a virus. A virus cannot reproduce itself. A virus cannot give you bacteria. So for life to be existing, for a cells to occur, it must come from another cell. So for one cells to become two is known as mitosis. Or for one cells to become two is known as meiosis. The only difference is meiosis for germ cells. Meiosis is for germ cells, egg and sperm. Mitosis for somatic cells, non-germ cells. And if you think about the germ cells, you want to get half of the chromosome from dad and half from mom. So the product of each germ cell should be haploid, 23 chromosome. Egg should have 23 chromosomes. Sperm should have 23 chromosomes. But when you talk about skin cell doesn't fertilize. So, it should have 46 chromosomes. All somatic cells, the end of somatic cell mitosis should be what? 46 chromosomes. And the end of meiosis should be 23 chromosomes. Okay, so I hope you understand. Meiosis, the end product is 23 chromosomes. Mitosis, 46 chromosomes. Okay. Somatic cells that go through mitosis cannot participate in fertilization. They will not be able to give you a fetus or a zygote or a baby for that matter. Somatic cells are identical, so they just replace the old cells. They don't give rise to like a huge human being, All right, I hope you understand it. But now, Mendelian law says that chromosome, during mitosis, the segregation of chromosome, if you have 2 pairs of each chromosome, and each cell is supposed to get 2 chromosomes, and then during S phase, this before prophase is what? Interphase, excellent. During the S phase of interphase, when cells duplicate its DNA, and then this chromosome has to segregate, this segregation is random. Random, random, and random. So that means we have 2 X chromosomes and 2 Y. You don't know which one are you going to get. We don't know which one are we going to get. In the case of sperm and egg, we don't know if a sperm is going to get a Y chromosome or X. Remember, egg or sperm, actually, egg will always get X. Sperm, we don't know if it will get Y or X. It cannot have both at the same time because it's haploid. Okay? X and Y is considered the 23rd pair. 23rd pair. So let's do this. Now, all these chromosomes, randomly, they should align. I'll use the smaller one now. They will align in the center. X, X, Y, Y. Randomly. We don't know who will line up where. This is not programmed in a sense, like who should get what. It's random. Reassortment, arrangement. The term that's used is reassortment. Arrangement of chromosome in the center of this plate called metaphase is random. And of course, then these things have to be attached by what? Spindle fibers. Spindle fibers by their centromere. Let's say this spindle fiber, randomly, first one will attach, it will pull it this way. And maybe this one will pull it this way. And maybe this one will pull it this way. And this one this way. At the end, there's even distribution of what? Of chromosome. Half chromosome is supposed to go to one side and half to the other side. Okay? So, you've seen this chromosome, and then we've seen this chromosome, and then this chromosome, and then this chromosome, and then this X, and then this X, and here's Y, and here's Y. So at the end, we segregate them. It's almost like, me personally, I always think of, when you play pickup sports. When you play pickup sports, you take the group of students or friends, let's say 20 of them, you make two captains, and you line them all in the center, in one big line. And then you say, you go this way, you go this way, you go this way, you go this way, randomly. That nobody has all the best players or nobody has all the worst players, right? So just 1, 2, 1, 2, 1, 2, 1, 2. At least in gym, teachers would do that, just to be fair. So assortment is random. At the end, you have to segregate this chromosome to each side. And then the cell will divide in half, and you'll get 2 cells. So each will get 46 chromosomes. Each will get 46 chromosomes, each cell. In this case, this is a male cell, so obviously it will be XY. Yeah.
I thought it sister chromosomes like two apart, so that it's one here, one there, but there's still one on each side.
This thing is almost like I'm sitting in Iraq and there's planes on top of me.
I thought it's sister chromosomes that flew apart at the centromere.
Mm-hmm.
But the way you were saying is like there's just one on this side and one down this side. Are you saying it's even?
Yeah, chromosomes, it is what you said is correct. But sister chromatid actually line up more in meiosis and crossover. In mitosis, there's no crossing over. They can align in the center, but still, it's random. Yeah. Okay? All right. So at the end, you will get half. Remember, this is supposed to be 92 chromosome in here for human beings. And now here's still 92. But 46 on this side and 46 on this side. And then eventually you'll get 2 cells. For whatever reasons, if 2 chromosomes are not separated, let's say both chromosomes move to the same side, that's called non-disjunction. Okay. Let's say in this case, or let's say in the case of chromosome X. In the case of chromosome X, both X chromosomes are attached by the same spindle fiber. Let me use red. By the same spindle fiber. And they're pulled to the same side. That's called non-disjunction. 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 So now, one cell might get more X chromosome, the other one might get less. Okay. Very good.
Dr. Punnett, is it normal?
No. Whenever you have more than one copy, more than normal copy or less than normal copy, it's called disease. If you have too many X chromosomes, look, how many X chromosomes is a man supposed to have? 1. How many X chromosomes is a female supposed to have? 2. If a man has 2 X chromosomes, 1 Y, what do you call that condition? Klinefelter's. It's a disease. I'll discuss those diseases. So abnormal number of chromosomes is a huge problem.
So this is the reason for this disease?
Yeah. Yeah. Non-disjunction is the number 1 cause of Klinefelter's.
So what's the cause of non-disjunction? Why?
Very good. Somebody's curious. Let me give you an answer. As we age, what happens to, what is, spindle fibers are made of what? How about this? Let's start with that basic thing. Microtubule. Good job, Nina, and you too. Microtubules. Microtubule is supposed to assemble and deassemble, correct? Now, in order for that to happen, number one, you should have appropriate amount of ATP, and you should be able to produce enough tubulin. And tubulin is a protein dimer. It's coded on DNA itself, how to make that protein, correct? So with aging, what happens with genes? It becomes more inactive, and you cannot replicate them or you cannot transcribe them at the same rate. So that's one thing. And when we age, what happens to protein? They get older themselves. If you don't believe me, look at my skin compared to 10 years ago. Yeah. So proteins get old, they wear out. Now, when you have these spindle fibers made of microtubules and they're weak, just like when we age, we're weak. During mitosis, do you think these spindle fibers can break? Do you think they can misattach to the wrong chromosomes, two at once? Yeah. And that's what happens. Number one cause for non-disjunction is aging. Not of the baby, not of the fetus, because they haven't even started yet. We're talking about mom or dad, sperm or egg. It's the aging of sperm or egg.
Even.
Now, let me go further. I know where you're going, my friend. Eggs, because females are born with eggs, set number of eggs. Sperms are produced every minute. Sperms are produced every minute. So maternal age is more likely. More likely. Maternal age is a strong, strong correlation with Down syndrome or trisomies or monosomies. Maternal age, number one. But paternal age is also associated. That means the sperm, even though you produce them every day, as you age, your ability to produce the same healthy sperm? No. You can no longer produce the same healthy sperm as you used to. Okay? And that's why, at least in this country, if you have noticed, you know, females, as soon as they reach over 25, 26, they want to have children for the same reason. Because they have the knowledge of, once you reach 30, there's a natural trend in America that all females are supposed to have kids before age of 30 or 29. Because of the risk of Down syndrome and trisomies and monosomies increase by far. Doesn't mean you will get it, but it's there. Okay? And a lot of other defects. So females more because eggs, you're born with a set number of eggs. And then they can age. They will age. The best time to have children is 22, 23 for both genders. Best time. Healthy children. And of course, when you have them, I have a friend, he had children when he was, 21, 22, and they're all older. He plays with them, and my children, I'm old and I have children now. One thing, having children early is good when you know you will have the energy to deal with it. And as you age, you lose energy. Yeah. But it's up to you guys, because these days people have different mentality and ideology. I prefer to have children. I wish I could have it 10 years ago. I didn't get married then, so. Anyhow, there you go. So why I wanted to do this is simply random reassortment. I did this diagram just for one purpose. Ahmed, don't laugh, don't smile at me. You better get married tomorrow. I have a girl for you. Yeah.
Oh, you're lucky.
Ahmed's lucky. Okay. What nationality, Ahmed? Ukrainians?
Yeah.
Got it. Okay. So the whole time I want to spend on this is only one thing. Random reassortment. Random reassortment. Random reassortment. Please remember that. Segregation of chromosome during anaphase is random. It's not planned. And this will help us determine today, when we get genes, is it random or is it programmed? It's random. Okay? So you don't know whether you will get the disease gene or not. Then we have to use statistics. What is the likelihood of getting that gene? Since it is a random phenomena, you know, it's not set, then we have to use statistics. What is the likelihood of getting a gene? So we're going to do that. So all I want to make sure, please pay attention to prophase. The whole idea here is random reassortment, random division of chromosome. Okay? You don't get to pick. You don't get to pick. You don't get to pick. If you had this ability to pick, I mean, what? Nobody will have diseased children. Or they won't even, if they know that they have this. For example, nowadays there's counseling done, right, for cystic fibrosis, sickle cell disease, and things like that. And you know, if you have if a family has a disease, a recessive disease, in their family running, they're supposed to do genetic counseling, especially if they're getting married from the same ethnic group. They're supposed to do that. And then if the physician knows, then they're supposed to ask. By law, physicians are required nowadays to ask about, I think, guns, smoking, and genetic defects. But not that you should ask, you should actually ask them. I mean, it takes time for all these laws to be implemented, right? For people to become adapted. So regardless, when you do those things, when you're getting married, or you have your children getting married, or your sisters or your brothers, it's a good idea, because you want to have healthy children. You want to have healthy children, for that matter. So genetic counseling is a common thing, at least in North America. Very common thing. Okay? All right, so all of this, you know. I'm not concerned about this. Only one thing. Please know that reassortment and division of chromosome is random and random and random. Okay, now we can go somewhere. Now we can start. All right, let's say, so symbol for mom, female is this. And the symbol for male is this. If I put a dot in the center of this, that means pregnant female. Okay? So, let's say, now do you know any disease? Huntington. Huntington disease, I'll put H. So it's on chromosome number 4. Okay, chromosome number 4. And let's say, so that's female, she should have X chromosomes, 2 of them, and 2 chromosome number 4. Male should have 1 Y, 1 X, and 2 chromosome number 4. Just for simplicity. Okay? Huntington is autosomal dominant. Autosomal dominant. Okay, it's trinucleotide repeat. Now let's say the gene, the disease gene, is located on chromosome number 4 of mom. I'll put the red. Now, here is the baby. Let's see, so remember these guys supposed to mate, right? So when they mix, let's just do this way. Here you go. They get married and they want to have children. Now, number one, let's focus from mom. Randomly, you will get 1 X chromosome. You don't know which one. In this case, both chromosome X are healthy, so you get 1 X chromosome. And you know what? Let me use a different color for paternal X chromosome. Can you guys see the green? Is the green visible? Yeah, it is. Yeah. Okay. So now, when you get X chromosome from mom, it doesn't matter which one you get, because they're both healthy. So let's get, you get X chromosome from mom. That's because it's random. You get this one or this one. Let me just put 1 and 2 on it, so that way we know what we're talking about. Or let's do this. You get the yellow X chromosome or the blue X chromosome from mom. It's random. We don't know. Regardless, no matter which one you get, it's both are healthy. It's okay. As far as the X chromosome is concerned, X and Y, the only thing you can get from mom is what? X chromosome. When it comes to dad, now it's a different phenomenon. You either get X or Y. So gender of the baby depends, or sex of the baby depends on who? The father. Okay, so sperm are either Y sperm or X sperm. Egg is always 1 X. Each egg will carry 1 X. But we're talking about a mature female. She's in diploid state. She should have 2 X chromosomes, but she can only donate 1. Remember, we talked about separation of chromosomes. It's random, and the product of meiosis is only haploid cells. 1 copy of each chromosome, not 2. We're talking about meiosis now. Okay, so now, let's say from dad you get the X chromosome. Isn't that 50-50, you get the X or Y? Yeah, so it's 50-50, you get the X or Y. And in this case, if you get the X, the likelihood of this baby is for sure female. Okay, so this baby, again, the first baby being female or male is 50-50. We don't know. But it depends on the man. It depends on the male. Okay. So that's one thing. Now let's talk about the disease part of it. Chromosome number 4 from dad, doesn't matter which one you get, because they're both healthy. So you get one chromosome number 4 from dad. But from mom, you can get the healthy or the diseased one. The healthy or the diseased one. Okay? Let's say we get the diseased one. Now the likelihood of the diseased one is 50%.
50 or 25.
50. Getting the diseased allele from the mom is 50%. Because she has two copies. You get the healthy or the diseased one, it's 50-50. Okay? This is just one child. Just one child. So now to do this, usually you will do Punnett Squares. And that would help you the most. I just want to show you on this for you to see that whether you get the diseased allele means a copy of a gene, or healthy allele is random. It's not set. It's not meant to be. It's random. It's random. Okay? So now let's do Punnett Squares. I included X and Y just to tell you the gender part of it. Okay? So when we write these genes, let's do this. Now we're talking about Huntington's disease. When we're talking about Huntington's disease, let's do a Punnett square here. There are 4 possibilities in terms of genotype. So dad, let's put on top, is chromosome number 4, healthy. Chromosome number 4, healthy. So these are chromosome of dad. And the maternal chromosome, the red one is, you know what, let me just do this. So dad has 2 healthy chromosomes, mom has 1 healthy chromosome, and 1 diseased. Red one is the diseased. Red one is diseased. When you do these Punnett squares, everything is random. You don't know which one you're going to get. You see in this line, this gene is supposed to go in both of these boxes. This green. If you're paying attention right now. Okay, so finish your writing. Finish your writing. In this case, mom is in this area, so this green gene, you see this one? It's supposed to go across. You're supposed to write across. And this one, you're supposed to write it across. And the paternal ones, you're supposed to write it downward. And let me do that. So green, or you know what, I'll give different color to mom. So look, mom's gene, here, and here. Red gene from mom, here, and here. Paternal. And let's make dad's blue so that way we can differentiate. Here, 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 here. So you have 4 different types of cells. But at least the top 2 are the same genotypically. These 2, both of them are the same. So 50% of the children are what? Healthy. Because they don't have the diseased gene. 50% of the children have a diseased allele. Allele is an abnormal copy of the gene. Okay. Now, because it's dominant disease, for dominant disease, you need only a single copy to express the disease. So you have phenotypically 50% of children are genotypically and phenotypically different. Okay. All right. So as long as you understand random reassortment. Now to understand this, we have to start with some terminologies. I just want to give you a brief overview of what we're going to talk about today. Let's talk about something known as phenotype. Phenotypes. To understand all these conditions, we have to know some terminology. Phenotype. Phenotype means appearance. Genotype is your DNA or genetic makeup. Genotype.
What's genotype?
Genotype is genetic makeup, the number of chromosomes. Okay. So genotype is your genetic makeup. Phenotype is your physical makeup.
I mean, in that case, not necessarily. I mean, if parents have like 4 children, not necessarily. No.
That's true.
Is that true?
Yeah. This is just possibilities. What are the possibilities that can come up? I'll tell you more about it once I finish. For example, if you have 2 parents, each have 2 genes, there's only 4 possibilities. Which one will come first? We don't know. It's random. So when you say when is the likelihood of the first child having a disease, Huntington's disease, 50-50. 50-50. Although there's 4 possibilities in that sense, because you can get the maternal gene and the paternal gene. Right? I'll show you one more time, as soon as we go over these. Phenotype is a physical appearance. Now, it's your physical appearance, that means disease as well. Symptoms, signs, defects. So when we talk about phenotypes, think of this as a disease. So you can have a healthy phenotype or a disease phenotype. Genotype is the makeup, genetic makeup. For example, for human, what's the genotype? 46, XX, or 46, XY. Okay? Okay. Dominant. Dominant trait, for it to phenotypically be expressed, you need only 1 copy. For dominant traits, for them to be expressed phenotypically, not genetically, phenotypically, you need only 1 copy. It could be characteristics, physical, it could be a disease. It doesn't matter. So in this case, we're going to talk about diseases. For dominant diseases, you need only 1 defective copy. Remember, we have 2 copies of each gene. For dominant disease, you need 1 copy of that gene. If you have 1 healthy, 1 diseased, you will still express the disease. Okay? So I want to make sure you understand. Dominant inheritance is done if you even get a single copy of a gene, you will still get the disease. Okay? Although we have 2 copies of each gene, right? Recessive. Recessive trait or a disease. If you have 1 copy normal and 1 diseased, you will not express anything. You're normal. Both copies must be diseased in order for us to express a disease, or both copies must be changed or mutated. Both chromosomes must be defective in order for us to express the disease. Recessive disease, both copies must be defective, must be mutated in order for a human being to express a disease, or anything else for that matter. So for recessive, you need 2 abnormal copies. For dominant, we need 1 abnormal copy. Okay? Does it make sense? Are we good? Okay. Yeah.
I have a question. So can a disease be dominant and recessive? Or
No.
is it just like certain diseases always dominant and certain diseases always recessive?
Now, there's a thing. There are certain diseases, but not conditions. So not a mutation. Mutation can either be dominant or recessive. But the result, so let's say we have a long gene. For example, let me give you this. Have you guys heard of severe? How many of you love Seinfeld? Did you watch that episode, The Bubble Boy? Can you tell me a little bit about it? What disease did that boy have? It's known as severe combined immune deficiency. Okay, so just to make a point for you. In this case, now you can have an enzyme deficient. Enzyme, it will lead to the same disease. Or you can have a receptor deficient. Same thing. Okay? The enzyme is responsible to produce the receptor. So at the end of the day, whether you cannot make the receptor or you have defective receptor, the result is the same. True or no? Now, enzymes are always inherited in the recessive fashion, and I'll tell you why that is. Let's write it down first. Structural protein. Structural protein, always dominant. Always dominant. Okay, structural protein. Now, to give you a real example, think about enzymes, construction workers. Construction workers. You know, to build a house, let's say a huge building in a certain time, let's say you need about 100 workers. 100 workers. 50 from factory A, 50 from factory B. And let's say factory A is on a strike, you still have 50 construction workers. So the product is decreased, the productivity is decreased. That means only you will have effects if you go under stress. So that's recessive. Recessive means you still have 50% of the product available. It will still do the job. So enzymes, because they do activity, they're functional protein. You know, they can break things and build things. As long as you have 50% of the enzyme, you still can do something with that. When it comes to dominant, think of dominant, not the construction workers, but the bricks themselves. If all the bricks are supposed to be square, and suddenly you have one brick only, and that is round, and you want to make a wall, the stability of the entire wall depends on it. The entire wall depends on one. So even though you have one copy defective, it affects all the normal copies, because it will not fit. So structural protein are usually dominant. So could you have the same disease as dominant as recessive? Yes, you can. Okay. Okay. So think of dominant disease are always structural protein. Remember we talked about, you know, mutations or defects are, it really doesn't matter unless the protein is affected. The end result is important. It's the protein product. If that becomes defective, then it's a disease. If there's a mutation in DNA, and there's a mutation in mRNA or ribosomes, and the result is silent mutation, it's no big deal. Silent mutation means the product is still the same, which is a protein. Protein didn't change. Amino acid sequence didn't change. As soon as you change amino acid sequence, or you change the protein structure, instead of having a water-soluble brick or amino acid, now you have lipid-soluble. It will change the whole thing. Think about it this way. If you want to make a boat, and this boat is made of paper that is water-soluble and water-insoluble, and the boat is supposed to be made of water-insoluble, so it doesn't dissolve in water, and suddenly you change one amino acid and make it water-soluble, there will be a hole in the boat and it will sink. Structural protein are always dominant. Dominant. No matter how many healthy copies you have, it won't affect it. One abnormal copy will affect everything. But for recessive disease, you must have both copies defective. And again, please remember, human beings have only 2 copies of each gene, not 3, not 4. Only 2 copies. That's natural. Too much, too little is a mutation in itself, or a problem. Okay, so are we okay with dominant and recessive diseases? Good. Now, we have total how many chromosomes? 46. How many pairs? 23. Out of these 23 pairs, so 23 from mom, 23 from dad, right? So that means you have one X and one Y chromosome, depending if it's XX or XY. You have 22 autosomes. 22 autosomes. Okay, 22 autosomes, and then you have X and Y chromosome. 22 chromosomes, they're not X and Y. And if you pair them, 22 times 2 is what? 44. You have 22 pairs of autosomes. They're known as autosomes. Autosome. So the first 22 chromosomes are known as autosome. Okay, and the 23rd chromosome is known as the X chromosome. Or sex chromosome. So 22 autosomes, and the 23rd chromosome is known as sex chromosome. It could be X or Y. So in general, we have 44 autosomes, and then we have 2 sex chromosomes. So we should have, every human being should have 22, or in total 44 autosome, or you can say 2 times 22, right? Or 2 pairs of 22 autosomes. But you have sex chromosomes, how many of them? 2. Now these sex chromosomes could be XX in one person, and another person could be XY, depending if it's male or female. Okay? Now further, we're going to identify diseases or mutations. Any mutation that's located on the first 22 pairs, what do you call them? Autosomal. See, if there's a mutation that's located on one of the autosomes, it's called autosomal disease. But if there's a gene that's located on the X chromosome, it's known as X-linked. It's linked to the X chromosome. For example,
Can be Y-linked.
now, very good. Y chromosome has only one gene on it, as far as we know, and that's for testes. Either you have it or you don't. Either you're male or you're not. So Y defect gives rise to hermaphrodites, for example. But there's no genes, no functional genes on Y chromosome. So that's why there's no disease linked to the Y chromosome. X chromosome has a lot of genes on them. So there's a lot of diseases linked to X chromosome. Hemophilia, for example. Hemophilia. Glucose 6-phosphate dehydrogenase, which is sort of hemolytic anemia. It's linked to the X chromosome. Duchenne muscular dystrophy. There's many diseases linked to X chromosome. So again, focus on, you have to understand, we have total of 46 chromosomes. 44 of them are autosomes. 2 of them are sex chromosome. And amongst the sex chromosome, only X has genes on them that codes for something. Y chromosome has no genes. The only gene it has is for testes. Okay? Yes, sir.
So does that mean that males are less likely to get X-linked diseases?
They're more likely. I'll tell you why. Okay? Okay. Men are unlikely to give X-linked disease to their son. Impossible. Think about it that way. But I'll come back to that. We're coming. I just want to make some sort of, basis for us to move forward. So we're okay with dominant, right? We're okay with recessive. We're okay with dominant and X-linked. And then we have phenotype and genotype. We're okay with those, all of them. Correct? Good. Now we also have one organelle, the crazy organelle, which has its own DNA. Mitochondria. So you can have mitochondrial diseases, which does not follow this pattern. What was the guy who discovered peas and stuff like that? Mendel. Yeah, mitochondria doesn't follow his Mendelian inheritance. Okay. And then of course, sometimes multiple things can be involved. We'll discuss that too. But for now, just know that when we talk about diseases, it could be dominant. Now, you can have autosomal dominant. Let's combine the two. You can have autosomal dominant, or you can have autosomal recessive. Same thing, X-linked dominant, X-linked recessive. Okay, so, but if you don't understand this, it will not make sense further. The least you have to know is what does dominant mean, what does recessive mean, what does autosomal mean, and what does X-linked mean, what does mitochondrial mean. You have to know those. Okay, you have to know this. And what is genotype, what's phenotype. And the core of all of this is, again and again, something so simple that you were taught in high school. Random rearrangement of chromosome, or reassortment, or segregation of chromosome during anaphase. Extremely important. Okay? All right. I can't believe we spent 1 hour on that, but we did.